Good afternoon, everyone. As Rob's mentioned, my name is Phileas Daisley-Guest, and it's my pleasure today to be moderating this exciting colloquium on ecological research with the Wolfram Language. We're fortunate to have with us an excellent group of speakers who have each made notable strides in using computation in Wolfram Language to advance our understanding of ecological systems. And our first speaker today is Gareth Russell. Gareth is an associate professor of ecology in the Federated Department of Biological Sciences at the New Jersey Institute of Technology. His presentation today focuses on animal movements in the Wolfram language. We're also joined by Zhu Feng, who is a PhD student at Boston University. Her presentation will focus on the effects of emergent competition on the properties of multi-trophic ecosystems, including the switch to top-down versus bottom-up regimes in the ecosystem dynamics. Our third speaker, Matteo Rizzuto, is a postdoctoral associate at Yale University. Matteo's presentation challenges us to rethink how animals shape our world and explores their roles in shaping biotic and abiotic Earth system processes in their environments, including in nutrient cycling and carbon capture. And then last but certainly not least, we're privileged to have Professor Jerome Goddard II from Auburn University at Montgomery. His research on predator-induced prey dispersal brings to light some interesting and surprising aspects of predator impacts on dispersal, and today he will introduce us to these and the underlying mathematics involved. Of course, we're very grateful to our speakers for accepting our invitation to this event and excited to hear from them about their great work and how computational analyses using Wolfram Language fits into their research. Each speaker will present for around 20 minutes, and we will have 5 minutes in between presentations for audience questions. So with that said, I'd like to extend a warm virtual welcome to our first speaker, Professor Gareth Russell.
This is work that was done by myself and my graduate student, Maggie Wisniewska, who graduated recently. And by the way, this research that I'm talking about today, it has been accepted for publication. It'll be out later this year, but it's not out quite yet. Okay, so I've been using the Wolfram language to study animal movements in general. And Maggie and I applied these tools to try to understand how elephants move in relation to their physical and social environment. Maggie previously had spent time in Namibia studying elephants in the field. And when she became my PhD student, she wanted to keep doing that. So we moved into a sort of a computational view of what she had been doing previously. What I mean by the physical and social environment is things like vegetation and water. And the social environment means other elephants. There are many elephants in this location that I'm about to tell you about, and they react to each other as well as to their ecological environment. So we are here, we are actually looking at Etosha National Park in Namibia. So here is an overview, and if you can see my cursor, Etosha is this sort of green area. The national park is this green area at the top. You can see that it has an unusual feature in it, which is this great salt pan. So if we zoom into the park, and this is a composite of satellite images from various times, you can see this large area, which occasionally has water in it, but most of the time is just dry and salty. And it's surrounded by arid savanna. And to the north, although you can't see it easy on this picture, there are some villages and things like that as well. And today, the data that I'm going to talk about come from the eastern region of the park. Here, we're going to focus just on this area, although there are elephants around the entire park. We have data from this area. Okay, so what data do we have to answer these questions? So these elephants are wearing GPS collars. The data I'm going to show you today are from 5 male elephants. There are data from more elephants, including female elephants, but we're looking at these 5. And these collars are getting a GPS fix every 15 minutes. And they were on these elephants for around 2 years. We also know where certain things are in this landscape. We know where permanent water holes are. And there are a few other point features that I will include in the analysis. And then in order to understand vegetation, we are also using NDVI, which is a measure of vegetation productivity, which is a product derived from MODIS satellite imagery. And that comes in at 250 meters of spatial resolution. And there's new imagery every 2 weeks. So it tracks the change in vegetation greenness over time. To do all this, I wrote a package. And here you see the top of it. And as you can see by looking at it, it's it's completely unannotated because after it says usage, there's just two sets of quote marks where there should be lots more information. This is not yet a Packlet. I know that Phileas has been encouraging me to try to turn some of these things into Packlets, which I do intend to do. But right now, it's a bunch of functions that all load in all at once, and I know what they do because I wrote them. But that's what we'll be using for these analyses. So it's meant to be a comprehensive package for analyzing animal movements. Okay. There are a few things we need to set up, and I'm going to take my life into my hands. We're actually going to evaluate some things live here. I know I probably shouldn't do that, but I'm going to do it anyway because I like to live on the edge. There's some information at the top about the raw data that we were provided with, such as where is the actual elephant's identification, where are the latitude and longitude columns and things like that. And then the second set are parameters that are needed at different times during the analysis, and it's easier to specify them all at once up front. I'm not going to go through all of them and what they mean, but these are just things needed to run that package that one needs to specify. I will mention the working projection. Because we're going to be measuring distances on the ground, we need something approximating an orthographic projection, and so we're going to use the standard UTM projection for our data. All right, so we need some landscape data before we look at the elephant data. We have a list of where the water holes are, and we have a list of FVAs. It doesn't matter for these purposes what they are exactly. They are another kind of point feature that elephants may be responding to. And what we're doing here is taking just the coordinates of these things and bundling them up with a specification about the projection and a name. And this name will be used throughout the analyses to identify this particular feature. So the point data, the coordinates, plus the projection, plus the name are something that we call a feature. And so we load both of these in. These are not very large files. And then for the NDVI was downloaded previously. It's a series of large data arrays. And what I had done previously is we stack them all together into a three-dimensional data array and then save them out using dump save in Mathematica so that we could load them in again very quickly. And here, again, we load in the raw array, which is this thing called NDVI stack, and then we associate with that a projection. So this is the projection of the NDVI data itself. We also specify the actual coordinates of the corners of it, and also the range of dates over which it occurs. And also we specify how we're going to resample it if we need to resample, and we give it a name, and we bundle all these things up into, again, what we call a feature, which is the NDVIF, standing for feature. Okay. And now for the actual movement data. So we're going to load this in. So this is sort of the original data set, and we load it in as a data set. You can see there's an index of observations. The elephants have names. There's 5 elephants, and this unit ID is actually the code that's given to each elephant. There's a time, a latitude, a longitude, and then a few other bits of information that the GPS collars record as well that we will not be using at this particular time. So now we start to use package functions. So there's a function called process tracks that essentially simplifies down these data to just the stuff that we need for the actual analysis. And it converts the latitude and longitude here into UTM coordinates, the working projection, because that's what we're going to need going forwards. So we've processed all those data. How many data are there? So we're looking at 350,000 lines. So there's 5 elephants and about 70,000 observations per elephant, and they're all in one sort of long data set. So part of this is a sort of a big, well, in ecology at least, is a big data issue here. And here is a nice map we made of those data, again, using Mathematica. And I have a sort of a 3x3 foot version of this on my wall. It looks very, very nice. Every movement for every elephant. And you can probably tell where the water holes are. And you can also tell where there are roads and tracks and things like that, because elephants like to walk along roads when they're there, because they're smooth and easy to walk along. So this is just a visualization of the data. So what do we have to do? So what we're doing here is we're taking one elephant and we're making it what we call the focal elephant. And then we are extracting the locations of the other 4 elephants and lining them up in time against the fixes of the focal elephant. So we know where each of the other 4 elephants was at the time when we knew where the first elephant was. In this case, it's elephant 264. So we have each time we're extending the data set that we already have. So now instead of locations, we actually have movements. So there is a time 1 and a time 2. Right, so this is now a complete movement segment, and we have the locations, the start and the end point of those movements. We know the time interval, which was 900 seconds, which is 15 minutes. We know how far the elephant moved, which is 300 meters, a little bit more than that. Okay, and we have the coordinates of the other 4 elephants at that time, which we'll be using later on when we try to understand how they are interacting with each other. All of these data sets, by the way, are showing the very first and the very last row of the data set. The reason there is no other elephants listed in the last row of the data set for this one is that the collar for elephant 264 ran longer than the others. So the very last fixes for 264, there were no other elephant data. And we'll be filtering those out in a minute. So we now have our data organized around one particular elephant's locations. The next thing we're going to do is filter the data a little bit. So we're starting with around 70,000 movements. We filter out sort of outliers. There's various qualities which I won't go into as to what filters we apply to the data. But when we're done, we've gone down to around 40,000 movements for that particular elephant. And now there are no lines without information in them. Next, we're going to add null movements. So what that means is for every elephant's movement where it has a start and an endpoint, we're going to add 30 randomly chosen alternative endpoints within a radius. In this case, it's a 300-meter radius, which is about as far as an elephant really ever moves in that distance. So now each line in our original data set becomes 31 lines. The first line, the one labeled data, that is the original observation, and the next 30 are sort of random movements that an elephant could have made but didn't. And this is the essence of the analysis, as you'll see in a minute. So now our data set is 1,230,000 lines long, because we've made it essentially 31 times as long as it was. And here is just a zoom in showing movement segments in red, and at the end of each movement segment there is a cloud of 30 points around that location, which the elephant could have moved to but didn't. All right, next we're going to calculate the distance from each elephant to each of its neighboring elephants. And so we do that. That takes a little while because we're not just doing it for each elephant's movement, but for all the 30 random movements that it could have taken as well. So these distances to neighbors are going to become predictors in our analysis, because we're going to ask, like, is a given elephant, in this case 264, is he walking towards? Is he trying to be near another elephant? Is he trying to stay away from another elephant? These are the questions that we're going to be asking. This one takes a little bit to run because it is running on over a million locations. In fact, while it runs, I think we can probably just go forward to the next slide. Or maybe we can't. Yes, we can. Okay, I see what's happened. So I've never run a slideshow before. It's finished. Okay, good. So now that we know, I'll just show you this. You can see now that as well as the locations, we actually know how far each elephant was away from the one that we're looking at. So this thing that ends in D is the distance to that nearest elephant. And now what we're going to do is go back to our environmental data, so our waterhole locations, our other locations, and our NDVI, okay? And we are going to sample from those features. So for the waterholes and the other point features, we are, for each elephant's location and all the null possible endpoints, we're calculating the distance from a waterhole, the distance from the other features. And for the NDVI, we're calculating what the NDVI value was at that location at that particular time. This is remarkably fast, actually, especially the NDVI sampling, because behind the scenes, we're using image and image 3D objects to represent arrays, and then we can use image value and you can sample 1 million points from a 3D array. If you convert it into an image 3D object, you can use image value and sample in just a few seconds. You can get 1 million samples, which is really very, very powerful indeed. So at the end here, you'll see, for example, these are the NDVI values for all every observation, all 1,200,000 observations, and these are the distances to waterholes and things like that. Now, just a bit of tidying up, we decide what analysis we want to actually do. In order to make the fitting process work better, we're going to scale our data to have approximately the same sort of range, same sort of ranges. And then at the end, we're going to jump back out of data set and just make it into a normal sort of nested list type array here. So again, you see the first and the last line in this format. So this is the format that we then use for our analyses. So this has all been data processing and sorting out. Okay, so what are we actually doing? Once we have this data set, we are fitting a statistical model called conditional logistic regression with a modification. So conditional logistic regression is actually this part. And what this says is, so in this case, J is the location that an elephant moved to in the actual data set. And X are the properties of that. So they're the NDVI value, distance from water, distance from each of the neighbors. And the beta term is a vector of parameters. Okay, so this whole term up here represents the quality, okay, of that particular location, the perceived quality of that location, based on its underlying properties. And then the denominator is the sum of the perceived qualities of that location and all the random locations. So all 31 random locations. And so this whole term is the relative quality of where the elephant moved to relative to where it could have moved to. And this is treated as a probability of that movement. It's a number between 0 and 1. The thing that we added here is that sometimes elephants move in response to things that are not in our data set, right? They could suddenly be spooked by lions, all kinds of things that we actually don't have information to. And they make movements that are simply, from our perspective, essentially random. And so S is the probability that an elephant makes what we call a random movement that's not predicted by our predictors. And if it does that, then all 31 locations are equally likely to be hit because that's a sort of a random occurrence. So this whole equation says if the elephant makes a random movement, then the probability of any location is 1 in 31. And if it doesn't make a random movement, then this tells us the probability that it'll be found at different, any one of those 31 locations, or specifically at the one that it actually did go to. So we have some functions that basically do this. So this is calculating the log likelihood, so the log of PM for just one movement. And these are just arbitrary parameters to show the function working. And this calculates it for all of the movements in the data set. So it's a very small number, right? Because that's a log number. And then we fit it using just actually find maximum, which in this case works very well. So here we're fitting a model, and this function, conditional logistic EML, is basically designed to look like Wolfram's standard fitting functions, where you specify a model and then you specify a list of parameters, and it goes ahead and it fits that. And what we actually do is we specify a maximum possible model complexity, and then we go through and we fit every possible subset model and create this list of sort of all possible models. And this is just a simple example that runs in a few seconds using a subset of the data. It actually takes quite a long time because we're using a very large number of movements, and there's a large number of predictors, and we also allow our predictors to have squared forms, like NDVI squared, for reasons that you will see in a minute. So in the end, there's 128 possible models of varying complexity, and we fit all of them, and then we sort through them using AIC. So down here, I put this code. I commented it out so I don't accidentally run it during the presentation itself. All right, my slide doesn't want to go forwards. Hold on one second. Okay. So once we fit a bunch of models, we use AIC, Akaike's information criterion, to rank them and calculate importance values for all the parameters. So this just shows a sort of toy example of ranking the models. In this case, the simplest possible model was the best one. And then these importance values describe the relative impact that different parameters have across all possible model subsets. So parameters that are in the best models have high importance, and parameters that are in poorly fitting models have low importance values. Okay, and we repeat the process for every elephant as a focal individual, and we do it for different regions of the park and for different seasons, wet and dry. So during the whole process of analysis, we're doing 38,400 model fits, although it's all automated and it just chunks away in the background. And it just takes a few hours to do that. So you can wake up in the morning and have the results of your analysis, which is nice. Once it's done, we need some graphical magic because this is quite a complicated analysis. And so after a lot of wrangling, we decided to make outputs that look like this. So on the left, we have a map showing that right now we're only analyzing movements that are near waterholes. So we want to know how elephants behave when they're near waterholes and how they behave when they're not near waterholes. And then to the right, these graphics show how elephants respond to the locations of waterholes and to the vegetation surrounding the waterholes and to the distance to this other point feature, which I'm not going to spend a lot of time talking about now. The curves show the shape of the fitted parameter function for the best model in each fit. Oh, and I should have mentioned this. We do 10 separate fits of different subsamples of the movements in order to check for model consistency, right? So our models should basically give the same result no matter what subset of data we collect, and they do. And so in these figures, the colors, the background colors, okay, correspond to these importance values. All right, and because there are 10 importance values from 10 subsets, we can calculate a mean and a sort of a confidence interval on the importance values. So the color of the circle is the mean importance, and the two triangles behind it show the sort of upper and lower confidence interval for the importance values. And so the dark blue ones show that in this case, when you're near a waterhole, believe it or not, and you'll be surprised to know this, distance to water is actually important. Distance to the other point features is somewhat important, and NDVI is actually not important at all when the animals are near waterholes. The shape of these functions, so because this is animal movement, if you see a U-shaped function, it means that the animal is making back and forth movements towards that feature, which is not surprising for the waterholes that animals do that. A descending function indicates, in this case, a descending function for the distance means that an animal is actually preferring it because it doesn't want to be a large distance away from it. Ascending functions indicate avoidance, and unimodal functions, which we don't really see any in this, except these ones are a little bit unimodal. These are NDVI ones. They indicate maintaining a roughly constant distance or preferring an intermediate value. The other thing we do, so that was for the physical parameters. Here we have how each elephant relates to the other elephants. Okay, so it's blank for itself, but this just shows how, for example, this shows how elephant 265 is impacting elephant 264, right, and so on. And then, having created these, we then take the importance values and we use them to construct a social network graph where the importance values become the weights of the social network graph. So here, for example, this top line shows us that AG264 is affected quite strongly by the location of AG268, a different elephant. Okay, and then in the social network graph, that becomes this thick line pointing in this direction, showing an influence, a direction of influence, and the strength of influence of elephant 268 to elephant 264. So turning these into social network graphs is something that we were very pleased to be able to do. And in the end, of course, we do this for different seasons and different regions of the park. And so ultimately what we're doing in this paper is we're showing how the elephants' social interactions change from season to season. And as elephants move away from waterholes and out into the wider landscape, their social interactions change, sometimes strongly, sometimes not so much. So to give you an example here, in the wet season, elephants 265 and 264 are constantly aware of each other's presence, okay? And they have U-shaped functions, so they constantly meet up and go away again and meet up and go away again. And they do that in the wet season in all regions of the park. They constantly are coming together and going away again. And that's fairly consistent. But then sometimes things change. So in the near water holes, AG268 pays attention to what 3 other elephants are doing quite strongly. But then when they go out into the rest of the park, that elephant stops paying attention to any of them except AG264. And so we can understand the dynamics of social interactions here. All right, so that's the paper itself. What makes the Wolfram language really good for this? It's fantastic to have all the geocomputation abilities to be able to work with landscape data and geocoordinates and all the movement data, just to be able to handle it in that geocomputational framework in one package is really, really cool. I mentioned this before, the image sampling is really good. Image value and pixel value are super fast. And so we load in the data, and if it's in any kind of an array or a dynamic array, we convert it behind the scenes into an image, and then we can use these to sample millions of locations in just a few seconds. And that's really cool. And of course, we can customize our statistics. So conditional logistic regression is a relatively common thing, and in other packages, there are ways to do that, built-in functions. But when we had to customize it with adding the random component, that would have been difficult to do, but because we would essentially hand-coded it ourselves, we were able to just add that in, and that was really nice as well. And the find maximum makes it fits really, really well, these data. And then, of course, visualization is super good in the Wolfram language. Geographics makes it easy to do, and we could also all those output graphics and the social network graphs and those things were sort of custom built to show our data to its best advantage. Using sort of graphics primitives, but it's all automated. There's a function that makes one of those figures now out of our results, and you can pull it all together. This is what we use for promotion. It's the map, and then I took an elephant picture that was taken by Maggie from when she was in Africa, and just used the Wolfram language to remove the background and then make it transparent and overlay it, so that we can really show off these data and these results to the very best advantage. A couple of things. So one of the things I think would be really cool to have in the language, which essentially I've coded in there, is something like a georaster object. So at the beginning, I took like the MODIS data and bundled it up with information about its projection and all those kinds of things. I think having a bundle like that, which is named, right, and is called a georaster, would be a super useful thing to have built into the language itself. And then you could just sample it using a function called something like geovalue. And essentially that's what I'm doing, but I haven't written a special built-in kind of object to do that with. But I think that would be a really useful thing for the language to have. And the other really cool thing would be that georasters should be usable as geographics primitives. And I've told Phileas this as well. You should be able to take a georaster and just drop it into a geographics and have it appear in the right place, projected into the right projection, depending on what that graphic has. And then users could contribute to a sort of a georaster library that other people could download as well. I think that would be super great. And I'll end it there. So thank you for listening. And if you ever want to contact me after the fact, there's my email address. Thank you.
Thank you very much, Gareth. That was really interesting. And while we're looking through some of the questions that are coming in, I just wanted to ask you, how might the insights from this work inform conservation efforts, in your opinion? Are there any obvious ways that this could impact conservation and environmental management efforts?
Yeah. So once you have made a model like this, once you understand what elephants' preferences are, and those what do they like and what do they don't like, right? What do they gravitate towards and what do they tend to avoid? You can take the model and you can take a new landscape when you haven't seen before, and you can actually assess it qualitatively, because we have these functions that apply to the underlying variables. I can take a new landscape, apply the fitted functions, and actually measure the quality of each pixel, okay, from an elephant's perspective. So this allows us to do things like examine which areas are better or worse for elephants. If something has happened, for example, humans have change the landscape, or there is some proposed change to the landscape, we can sort of evaluate it in terms of, is this going to make the landscape better or worse for elephants? It could be better. It could be a restoration effort or something along those lines. So it can guide the way that we manage landscapes. It also allows us potentially to predict where elephants are likely to move. This national park has a fence around it, so the elephants are sort of constrained within it. But many protected areas in Africa they're not fenced. Elephants move in and out, and it's of interest to know where elephants are likely to go, because then we can try to minimize things like human-elephant conflict, which is a big issue in many parts of Africa, and things like that. So all of this was done with an underlying conservation goal. That's what was motivating us.
We have a question from Wissam, which is how consistent were the human, quote unquote, field observations of Maggie with what you collected and analyzed from the collars?
Oh yeah, so that's a great question. So the three big water holes that I showed, or you can see on this picture right now, you can see where they are. They actually have permanent field stations there. So they have a tower where researchers regularly observe what goes on at the water hole. And so we already had a good idea about how these elephants interact when they're right at the water hole. And there is a dominance hierarchy. The first two elephants, 264 and 265, for instance, are large male elephants, bulls in their prime. And then we have a juvenile, a sort of a medium-age elephant, and we have an older elephant who's sort of past his prime. And so it was quite well understood what they do around the water holes. And what we observed from our data is consistent with that. So one of the things is that we sort of validated our model by comparing it with the observations around the water holes. And it's broadly the same. We see the same kinds of dominance hierarchy in movements as you can see when you just watch them behave. But what we're able to do, because we have the collar data, is we're able to see whether or not that same kind of hierarchical dominance continues when elephants leave the water holes, especially in the wet season when they spend less time at the water holes and they go wandering through the landscape. Are they still interacting the same way? Is there still a dominance hierarchy? And the interesting thing is that the answer is it does change, but there are still interactions that occur even when elephants are tens of kilometers apart. Elephants generally know where each other is. They can communicate over very long distances. They seem aware of each other's location. And the interesting thing is that when in the wet season, when there is less need to be drinking, in other words, when there's less water stress, elephants interact with each other more. And in the dry season, when they're really short of water, they tend to behave a little bit better. So you can sort of see this in this figure that I'm showing right now. The right-hand set of three those are the wet season interactions, and especially the wet season near water holes. They come to water holes in the wet season, but they spend a lot of time interacting with each other instead of just going down to the water to drink. So yes, it does compare with what we know about water hole behavior, but it also lets us look further afield.
Well, thank you very much for answering these questions. I think it's time for our next presentation. So
yes.
well, please welcome Zhu Jie for our Zhu Jie, you have the floor.
So I'm a physics grad student in Boston University. In our group, we use a lot of statistical physics methods to study an ecosystem. And my work will be about how emergent competition shapes the ecological properties of multi-trophic ecosystem. So to start with, our motivation is ecosystems are really complex in general. It includes many species interacting in one environment, and the interaction are so uniform, non-uniform and strong. And there's a lot of flow of energy and nutrients, which makes everything very hard in general. But fortunately, there's always, often, an approximate structure that's either called food chain or trophic levels, which means if you organize the systems, you can see they are structured in a hierarchical way where the species in one level will interact only with the species of the level above it or below it. For example, wolf here will interact with only the hamster rabbit and lions and owls, but nothing else. So the problem with the existing models of these trophic ecosystems is that it lacks the full picture. So classically, when ecologists study a food chain, they will effectively make a one level as almost a one species. So, for example, here you have lion, wolf, and rabbits, and effectively there's only one species in one level, and there's no diversity in each level. And there's another type of models that's called MacArthur consumer resource models. In that model, the species are categorized into consumers and resources, and there's multiple species in each level, but there's no vertical diversity. So our motivation is, what if we combine them to study multiple levels with diversity in each level? So to start with, I'll introduce you what the MacArthur consumer resource models are. So it's a model that consists of two sets of ordinary differential equations. So one equation will correspond to one species. For example, if there's 5 species in total, there will be 5 equations. And for dynamics of consumers, it will include a growth by eating, that's the interaction with resources, and it contains a natural death rate. And for resources, the only difference, it has logistic growth. And logistic growth means it will either be zero or saturated at a fixed population, which is K here. We call it carrying capacity. And that logistic growth is due to some environmental bounds, such as sunlight and water. And similarly, it has an interaction with the consumer, but it's a negative sign. So it's the death by feeding term. So what this model teaches us is the competition is emergent. And what I mean by that is, if you look at a classic example of two consumers fighting for one resource, as time goes by in this model, you will see only one species that's better at consuming this grass is surviving. And it's intuitively true. If there's nothing else that then the grass is determining how much they're growing. But if you have two resources, it is possible that the hamster and rabbit could each be good at one thing. Maybe rabbit is better at grass and hamster is better than carrot. Then they are possible to coexist in a ecosystem steadily. So what we did is we generalized this consumer resource model so that there is an intermediate. Oh, I think the equations are gone, but it's fine because I'll show you in the mathematical notebook. So let me just directly go to the notebook so that you can see the equations. Let me do that. So could everyone see my mathematical notebook?
Yes.
Yes.
Great, thank you so much. So these are the three sets of equations. It's almost the same as before, but only for the intermediate levels to have both a interaction with the, for example, carnivores and the interaction with the resources, like the plants, for example. So in this type of model, if you simulate them, which I did in mathematical notebook, you will see a typical dynamics looks like this. So you will see each line will represent one species, their abundance at that time. And as time go by, they will start to saturate at one point, and that's stably things will coexist. But a lot of things will also die due to extinctions. That you can see at 0. So below, I'm plotting the histogram of the final population of species. You can see for carnivores and herbivores, there's a big peak at 0, and that means species died. And how many species will die will be determined by everything else, like how many species initially there are, and also the all the parameters I choose from. And in this work, we often simplify things by looking at random ecosystems, which mean we draw a lot instead of choosing each parameters, like all the interaction between every species, we draw them from random distributions. So let me simulate this again. So this time, for example, you can see there's 6 species surviving in the carnivores. If I do it again, instantaneously, you can see this time there's only 4. And although on a small scale, you will see very different at each time, but as the system grows larger and larger, you will see some more consistent behavior. So let me go back to my slides. So in this model, the competition exclusion principle is generalized. So now again, if there's hamster and rabbits fighting for grass, on the left-hand side, if there's nothing else as before, only one species will survive. But on the right-hand side, if this time introduce a wolf, and this time maybe there will be one species better at hiding from the wolf, and one species will better at eating the grass. So in mathematically, we can describe this as the number of herbivores will be less than the number of surviving plants and carnivores. And more generally, if you make this into larger and larger system, we can always say the number of species is less than the dimension of the niches. So what's new with our model? So in this model, what's most interestingly is there are 2 ways to control the middle level. So imagine you have a job to control the population of rabbits and hamsters. What can you do? You have 2 ways. One way, you could change the amount of sunlight, for example, and that will change the amount of plants they can eat and will change their population. Another type of method will be top-down control. So you control the population of wolf. For example, you can hunt for them. You can help their growing. So the classic ecological question is, which control is more significant for one ecosystem? And how does the environment and species diversity will affect these controls? So in our model, I can show you what bottom-up control looks like. So here, we can both change the death rate of carnivores, and we can change the energy flux to the plant that correspond to top-down and bottom-up control. And in this selected parameter range, we will see there's not much response for this orange line, which is the middle levels mean population. There's not much response for that. The line is always flat. But for changing the energy influx, you will see a big response. So we call this ecosystem primarily bottom-up controlled. And similarly, there are parameter range where you can see primarily top-down control. So here, if you look at left, when you change the death rate of carnivores, you will see a big response from the middle species. And on the right, this time, changing the sunlight doesn't change the middle level too much, and that's top-down control regime. So what we do theoretically to analyze these seemingly very complex models, like you will ask, so there's so many parameters, like it will grow so much exponentially with all the added species in the system. What if we are looking at a large system? And our answer is that actually a large system gives us a lot better intuition and a lot better systematic way to study their statistics. And that's because of central limit theorem. So if you look at this model, intuitively, you can see there's a lot of sums. That's the sum of interactions. And when there's a larger and larger ecosystem, you would expect this sum to be a sum of very many terms. Then, because central limit theorem tells us when you sum a lot of random variables, you should see a Gaussian. So that's the intuition behind why we use a mean field analysis, which is very common in physics research to study this model. So on the left-hand side, I'm plotting a large system's dynamics, where you can see the eventually saturates. And on the right-hand side is our cavity methods predictions. And I'll show you from Mathematica what it looks like. So here I'm writing down the final result of our derivation of what the cavity method, the mean field analysis give us. It will give us a set of 9 equations that's coupled with each other that includes the mean and variance of the abundance of each levels for example, the mean carnivore abundance, the mean herbivore abundance. So we don't care about the individual abundance now. We don't care about the statistics, and we can actually solve them with Mathematica. So here I'm defining the solver for these equations. And let me show you graphically visually what it looks like. So the dashed line here, the black dashed line here is the solution of the cavity method equations, solutions. And the histograms here are the simulation results. And let me show you when I increase the number of species in this system, how it looks like. So now we only have 10 carnivores, 11 herbivores, and 12 carnivores. Oh, sorry, 12 resources, that's a typo. When I increase the number of carnivores, you can see the histogram looks more and more similar to the prediction of the mean field analysis that I did. And that's because of central limit theorem. And also you can increase the number of repeats you do. That will also make the prediction more and more accurate. So going back to the slides, so mathematically, of course, we successfully predict the statistics, but also conceptually, there's a meaningful way to tell what's the cavity method told us. So it told us before we have a large system of individually interacting species, but we can also view it as a system of only three things, three levels, but they have now like a emergent competition. What I mean by that, concretely, is like if you look at the equations before, there's only interactions across levels. But on the right-hand side, after we do the cavity method to study the equation in a level perspective, we'll find there's a new term in the red circles. That is the what we call emergent competitions. That means before, even though you didn't put a direct interaction between species in the same level, although there's no regulating terms for consumers before, but now you have that. That's the same as a self-regulating term for the plant before. That's almost the same. That term only happened because of the interactions indirectly with other species by competitions for either food or hiding from consumers. So we actually got the formulas for how these competition terms looks like. And surprisingly, it's really simple in the way that all of them mostly depends on just one fraction. And that fraction is related to how the niche is packed in the ecosystem. For example, as I said before, in our model, the number of herbivores cannot exceed the number of carnivores plus the number of plants. That's mathematically true and also intuitively true for ecosystems. And you can see, for example, if we look at the competition for herbivores, it will be positively correlated with these fractions, which means if you want the maximum number of herbivores in this system, effectively, their competition in between each other is actually zero. And that's the ideal case that allowed them to have maximum number of herbivores. And in the denominator, similarly, the number of herbivores minus number of carnivores is zero when the number of carnivores is at its maximum because of the competition exclusion principle. Similarly, that will be infinite if you want no competitions between carnivores. So conceptually, even though in our model there was so many parameters, at least 12 here, you can actually simplify things, and you got 3 parameters three meaningful quantities of competition that primarily depends only on the species packing when the system is at steady states. And another result we got from the analysis is that if we look at the competition between herbivores, it actually consists of two parts. And one is competition for food, and one is the competition for avoiding predators. That's mathematically obvious when you look at the math. And that provides us an order parameter for measuring how much this intermediate level is affected by top-down versus bottom control. And actually, this time it's even more simple. If we write down the fraction of top-down control among all the competition experience for herbivores, you see this is actually just the number of predators divided by the number of the number of carnivores divided by the number of herbivores in the systems. And that's surprisingly very simple. So again, there's so many parameters in our model, but in the end, we got these simple parameters for describing how much top-down versus bottom-up in the system, only depending on the species packing of the system. And this is just to show this order parameter is actually agreeing with other parameters you could think of. For example, if you measure just the food, like all interactions between carnivores and herbivores versus all herbivores with plants, or you can also look at the, as before, you can look at the slope when you change the sunlight versus death rate of wolves, and both parameters kind of agrees. And finally, what's interesting for us, or maybe a lot of people in ecology, is how diversity and environment affect everything, including competition and how much top-down control versus bottom-up control you have in the systems. So here we are doing a numerical experiment. We're just changing the parameters in our systems, including the fraction of the ratios of species initially in the system, like how large the regional species pool is for each level, and can change, for example, the death rate of carnivores and energy influx to plants like sunlight. And so you can also change the variance of interactions, which is something new, and I think a lot of people didn't know how to do. And this is allowed by our cavity method, because that's the cavity method is a really fast way to get all the solutions, all the steady states of the system without doing simulations. So this small figures is how we how we vary all the parameters we are interested and observe the trend, and that's the summarize of the trend in the systems. And we compared these trends with the literature we find from ecological literatures, meaning they are either observations or hypotheses. And when you have time, I encourage you to look at them. They are very satisfying for me, at least. So in conclusion, we build a model for trophic levels, and we generalized the original just food chain model to these models with diversity in the levels. And we solved analytically by cavity method for the statistical properties of these models to get the emergent properties, including competition, and also the order parameters for top-down versus the bottom-up control. And finally, these properties is surprisingly mostly only depending on the species packing, like how many species eventually survive in the systems. So our paper is published in PLOS Computational Biology, and encourage you to look at that if you're interested, and feel free to contact me. So future work, we actually put only minimal structure of interaction here, and we want to, if we want to study a more specific ecosystem, we would need more structures. For example, the omnivore or decomposition, cross-feeding, or you can think of something like a specialist instead of generalist, which we actually assumed when we say, and we can model the system as a random system. And we are, of course, interested to look at experiment and data to predict response of actual ecosystems. For example, a fish, zooplankton algae system, or a phage bacteria host cell system, which is actually a very popular field right now. And of course, you can use these results to use in actual agriculture, conservation, and things like that. I'll thank everyone involved in this work. Pankaj Mehta is our group advisor. Jason Rocks and Robert Marston are co-authors of this paper. And I'll thank the funding. And also, I used some cartoons. They're cool, and they're from this free pick website. Thank you, everyone. Feel free to ask me any questions.
Thank you very much for that. You answered my first question for you in your next to last slide, which was about like follow-up development for this model. But Matteo asks, how does your model differentiate between different species in the same trophic level? For instance, what sets two herbivore species apart in the model? Could you tell us a little bit more
in
about
this
that?
model, the only thing that sets two species apart is how they interact with other species, and also their intrinsic death rates. So if I pull up my slides, go back to the equations. Let me go back to this. So each herbivore has several interaction parameters like CIQ here that tells you how much it interacts with the resources. And that's also like a fingerprint for the herbivores in our model. I hope this answered the questions.
Yes, thank you very much. Okay, so then in that case, I think it's time to pass the torch to Matteo.
So, well, thank you, Phyllis, and Robert, and everyone at Wolfram for inviting me. I'm very excited to be here presenting it about my work, and thanks for the presenters that went before me and for the ones that are coming after me. Your stuff is really cool. It's really excited to be featured amongst great scientists like yourselves. Today, I want to chat a little bit about some of the work that I've done at the end of my PhD and for my postdoc, looking at how animals shape our world, focusing in particular on ecosystems, nutrient cycling, and carbon capture. And just to give a bit of a roadmap for the rest of my talk, I'm just going to cover some ecosystem ecology basics that will help us through the rest of my presentation. Then I'm going to talk about why we study animals within the ecosystem framework and complement that with a real world example. And then look at how in my work I have tried to integrate animal ecology and behavior into ecosystem models to explore how animals move and shape nutrient cycling at multiple spatial scales, but also how can they contribute to the ability of an ecosystem to capture carbon from the atmosphere, and then have some conclusions and takeaways. And so I want to start by just looking at the word ecosystem for a second, because as many of you probably know, this is a composite word. It comes from Greek. And in particular, I want to focus on the first part, eco, which comes from ancient Greek oikos, meaning house. And when I think about an ecosystem and I think about an ecosystem in terms of the work that I'm doing, for me, it's very helpful to think about it as a house because our formal definition is that of a system formed by the interactions between organisms and their environment, linked through nutrient cycles and in energy flows. And I think that more formal definition really matches very well to the idea of a house, a place that has very clear confined boundaries, where multiple people exist together, interact among themselves and with their surroundings. And so I really like to keep this idea of a house in my mind as I think about how animals fit into ecosystems and how they shape ecosystems. And I think it's going to be helpful to keep it in mind as we progress through this talk today. I'm going to be focusing for the rest of my talk on nutrient cycling. And nutrient cycles, as pretty much everybody in this room will know, look a little bit like this. This is just a diagram that I copied from an online source. and it's very simple. It's built on a terrestrial ecosystem, and essentially what it shows is that nutrient cycling, nutrients move in the ecosystem powered by sunlight, which is this dashed line here, and move from plants to animals in the soil, the composers, the thritivores, and going back into the soil. Now, we have nutrient cycling in pretty much every single ecosystem in the world. However, most of our understanding of how nutrient cycling and biogeochemical cycles in general work and how they influence ecosystems and the organisms that live in them come from studying what plants and soil and decomposers do. We tend to ignore these larger animals that I have highlighted here, which are the focus of my work. And at this point, you may be wondering why should we study those in the context of ecosystems? Well, because as it's at this point public knowledge, we have a heating problem on this planet. We're heating our house quite strongly at this point. It's turning into a little bit of an oven for everybody living, including us. And we have a main culprit for what's going on. It is the carbon dioxide that we put out in the atmosphere, and that we have started putting out in the atmosphere in increasingly higher amounts from about 200 years ago onward. As people have started noticing what was going on, we have started developing strategies to capture carbon from the atmosphere and store it somewhere where it can cause no further harm. There is some examples of atmospheric carbon capture technology currently active in some countries in Northern Europe. I don't remember exactly if it's Finland or Iceland. The problem with these kind of technologies is that at this point, they're still very small. They're hard to scale up, and they still are not particularly efficient when it comes to the amount of energy that they require to produce any benefit. So there's been a lot of talk about nature-based climate change solutions. If you've ever heard anybody saying we need to plant 1 billion trees or 3 billion trees, that is what I'm talking about. Essentially leveraging what nature has created over millions of years of evolution to help us fight climate change. And there is empirical evidence and some more conceptual work being done now that shows that animals can play a very important role in helping us develop nature-based climate change solutions and tackle climate change. However, the traditional wisdom, the existing knowledge that has shaped biochemistry and our understanding of nutrient cycling rests on two main assumptions. The first one is that ecosystems are controlled from the bottom up when it comes to the cycling of nutrients. So that pyramid on the left, we're probably all familiar with from our high school ecology textbooks or chapter in our textbook. Large base made up by soil nutrients and plants, and very tiny top formed by animals, and larger animals, in particular mammals, tend to be a very tiny portion of the biomass of any given ecosystem. The second assumption that traditionally biochemistry rests upon is that what is more abundant in terms of biomass is more important for the cycling of nutrients. So effectively, we can safely ignore animals because there is so few of them compared to plants and microbes in the soil that they don't really matter. Or if they matter, they only matter as a compartment that removes biomass from the system rather than doing anything to shape how that biomass is formed and how the ecosystem is structured. This is not always the case, and I'm going to show you why. And the talk that just preceded me by Juje, I think, made a great point to show that that is not also always the case. The other reason why we may want to study how animals fit in ecosystems is that there is one animal that has changed how ecosystems look like worldwide, and that's us. So the infographics at the top come from an article in The Guardian from 2018, which was picking up the paper that was published in PNAS by Baron and colleagues in 2018, showing the distribution of biomass on Earth. And this paper was showing that of all of the mammals that exist on Earth, 96%, it's either us or the things that we eat, the livestock that we grow to eat and to nourish ourselves. Only 4% of wild mammals are left in terms of biomass. And when it comes to bird, things are a little bit better, but it's still scary because 70% of all the birds in the world are now chicken or other poultry that we use to eat, that we use to feed ourselves. And the rest 30% is wild, but it's still a far cry from how things may have looked like before we became the dominant species and started changing the ecosystems around us. So these changes to the makeup of the mammal group, the mammal biomass on Earth, are shaping how ecosystems work and function in the wild, and how they can help us, well, how they react to the climate change that we are enacting. And so I want to tell a story very briefly, looking at how animals do influence ecosystem properties and ecosystem functioning and biogeochemistry. And then I'm going to talk about how my work is looking at what they do and try to put a more conceptual, rigorous mathematical frameworks around it. Because most of what we have currently is empirical evidence, like the one I'm going to tell you now. So we're going to travel to Eastern Africa, to the Serengeti. If you've ever seen a David Attenborough documentary or any documentary on Netflix, you probably already know this story. This is the wildebeest migration in the Masai Mara and Serengeti and Ngorongoro areas. The wildebeest migrate seasonally following that red dashed line, and their migration follows seasonal rainfall that greens up the landscape as it moves. And now, the wildebeest are a key feature of this landscape. Thousands of people go there every year to see the mass migration, but we came very close to losing it forever. And we came very close to losing it forever because of a virus that in the late 19th century, early 20th century started decimating the wildebeest. Now, we should be almost thankful to the virus, because through it, we were able to understand how wildebeest shape the whole Serengeti ecosystem and start getting an inkling of how important animals are to biochemical cycles. But it is really the villain of this story, because by killing and decimating the wildebeest population, I think they went from more than 3 million to a few 100,000 in just the span of a few decades. What the virus did was remove herbivory from the landscape, essentially. And so plant biomass exploded. There was a lot of biomass of plants on the system, which meant a lot of wildfires, more intense, more frequent. And that led to the Serengeti for the first half of the 20th century being a massive source of carbon dioxide to the atmosphere, essentially helping us make the world warmer rather than helping us cooling it down. In the 60s, people noticed what was happening. They developed a vaccine. And from the 60s onwards, the wildebeest population started rebounding, which meant herbivory came back into play into the Serengeti ecosystem, which meant that our plant biomass was reduced. This had the indirect effect to also reduce wildfires in both frequency and intensity. So that today, at this point, 60 years later, the Serengeti ecosystem is a massive carbon dioxide sink, and it's able to capture enough carbon dioxide on a yearly basis to offset most, if not all, the emissions of Eastern Africa, which are the countries in green in the little map. So my work looks exactly at this kind of effect of animal presence and behavior on ecosystem functioning. And I use mathematical models developed in Mathematica and other softwares to explore how animals shape ecosystems, what does it mean for emerging properties of ecosystems like nutrient cycling, carbon capture, and whether we can leverage their help in fighting climate change. And so the kind of models I work with are very similar to the things that Juje, I hope I'm saying your name right, I apologize if I'm not, was showing before. These are MacArthur models, compartment models, process-based models, in which we have different compartments that represent different components of an ecosystem. So here, the yellow atom is not nuclear energy, it's the soil. And then we have the plant, the zebra, and the lion, showing primary producers, herbivores, and predators. And in a model that I'm going to talk about a little bit later in this talk, each of these is represented by equations, ordinary differential equations, like the one that I'm showing on the slide. In this case, this model that I will be talking about in a little bit tracked the content of both carbon and nitrogen in each of these compartments. So you have a couple of equations for each compartment, but otherwise it's usually just one equation for each compartment. And as I was talking earlier, in the wildebeest story, the wildebeest engaged in two important behaviors that I mentioned. One of them is moving, and the other one is foraging, which is also part of what I was very interested in Garrett's talk earlier, because that movement stuff was really, really interesting. So I want to talk to you about two recent papers that I published. One looking at how animals shape carbon cycling and rewire it inside the ecosystem, and the other one about how their movement influences nutrient flows between ecosystem, and that changes how both locally and regionally ecosystem function. And I'm going to start by this second one. And if you're interested, you can find the paper or the Figshare repository with the Mathematica notebook and additional code following the QR codes that I just put on the screen. So the question for this paper was essentially, how does animal movement shapes how nutrient flow in an ecosystem? And the idea for the paper came from looking at the study area for my PhD. I did my PhD in Newfoundland, which is a large island off the coast of Canada, and it's covered in the boreal forest. If you're not familiar with the boreal forest, it is one of, if not the single largest biome in the world. It's a giant belt of trees, mostly conifers, that spans from Newfoundland in Canada all the way to like Russia and Europe and Scandinavia, and it's one of the largest lungs of the world, together with the Amazon. And so the thing about the boreal forest is that because it's found at such a high latitude, it's a very nutrient poor ecosystem, and so animals really need to work a lot to find their food. And so we wanted to look at how an animal may move in a system where the landscape varied in its availability of food, like the grid that I just make appear on the slide. And so most of this ecosystem, this landscape, is not particularly good for food. There is almost no food available, and that's the gray. And some areas have food available, and that may change in its quality from low to high, or even just in its quantity from low to high, based on some characteristic of that area. And so if you're an animal, I was interested in the question of how do you decide where to start your movement, and then where do you go? Which ecosystem do you try to find? So I built a meta-ecosystem model, and that's simply a model that simulates the connection between two different ecosystems that are connected by either inorganic movement of nutrients at the bottom between those two orange boxes that I show here, or in our case, movement from the top. So consumers, the purple boxes, leave ecosystem 1 and enter that Q compartment, and I'm going to talk about what that is in a second, and then leave Q and enter ecosystem 2. Now, each ecosystem is described by a series of equations, and the equations capture the trophic relationship, those are the gold arrows, the recycling relationship, those are the dash arrows, the inputs and outputs at the bottom, the white and blue arrows, and the movement of consumers, the magenta and the light blue arrow. Q, it's something that we borrowed from the epidemiological literature. It's a disperser's pool, which essentially means that when the consumers leave ecosystem 1, we do not immediately assume that they are going to enter ecosystem 2, but they're going to spend some time outside ecosystem 1, and then after a little while, enter ecosystem 2 to sort of represent that time spent moving around in that part of the landscape that doesn't have any food, searching for a better place to forage. And use this model, we can simulate the movement of consumers in a few different scenarios. We pick two, one where consumers are moving from an area of high to an area of low resource availability, and that's on the left along gradient movement. And the other one was the opposite, moving from an area of low resource availability to an area of high resource availability, and that's against the gradient on the right. And so we plug this model into Mathematica, solve for equilibrium, explored local stability, calculated a bunch of different ecosystem metrics that you can find in the notebook, but I didn't have time to put on the slide. And then we run, I think, 10,000, if not more, simulations of the system and how it changed over time. And we focused on the effect of animal movement on three different properties of the ecosystem. The first was stock accumulation, so the accumulation of soil, plant, and animal biomass, productivity of each trophic level, and nutrient cycling operated by each trophic level. And I'm just going to focus on the nutrient cycling part because that's the interesting bit. I'm going to show you just one graph of results. On the y-axis, we have the log response ratio, which is essentially a measure of relative change. If something appears above 0, that means it's increased compared to like the baseline. And if it appears below 0, it means that it's decreased compared to the baseline. There are 3 panels, meaning the 2 ecosystems and then the overall larger meta-ecosystem scale. And our animals are always moving from ecosystem 1 to ecosystem 2, as the red arrow shows. And in yellow, you will see what happens when ecosystem 1 is the system with more nutrients, more resource available, and ecosystem 2 is the one that has less available resources. And in red, you're going to see the opposite, when ecosystem 1 is the system with less forage, and ecosystem 2 is the system with more forage. Hope that's clear. Let's go. So in ecosystem 1, we don't really see much of an effect of consumers, of animals moving on the ability of primary producers in moving nutrients in the system, in the nutrient flux of primary producers. As we would expect, when consumers are moving out of a rich ecosystem, we have a high nutrient flux mediated by them. When they're moving out of a system with low nutrient availability, we have low nutrient flux mediated by them. What's interesting is what's happening in ecosystem 2. Here, when consumers are moving towards a resource-poor ecosystem, nutrient flux, so the ability of those 2 trophic compartments to cycle nutrients in that system, decreases with consumer movement. And the opposite is true. So we see an increase in nutrient flux with consumer movement when the consumers are moving towards an area of high resource or food availability. At the regional scale, the meta-ecosystem, we see a more subdued response, but more similar to what's happening in ecosystem 2 than to what's happening in ecosystem 1. So what this tells us is that animal movement has the ability to shape where nutrients accumulate in the ecosystem and in the regional, in the whole landscape, potentially either compounding landscape heterogeneity or diminishing it, depending on how they're moving, whether it's along or against the gradient of resource availability between the different ecosystems. Now, this is important because animals do move. They move a lot. They move pretty much all the time, either to forage or seasonal migrations, life cycle migrations, dispersal to colonize new habitats. They move a lot. Mass migrations are the best and most impressive of these movements. The best known and most impressive of these movements, they travel thousands of kilometers. Other than the wildebeest example here, I'm showing the Canada geese, the caribous in North America, the humpback whales in the Atlantic. These are all migratory animals. So we don't really know how important the transfer of nutrients that they mediate are for the ecosystem that they visit during these migrations. But through the model that I developed, and the work that others are doing in this meta-ecosystem theory framework, we could potentially start getting at that, at those results. And this is important when we look at things that are a bit more important for climate change, such as the ability of an ecosystem to capture carbon. So moving to the next paper that I want to talk about, again, you can follow those QR codes to find the paper itself and the code. We wanted to look at how animals change and an ecosystem ability to capture carbon. Again, we started from the same general model with 4 compartments: soil, plants, herbivores, and predators. This is the one that I showed you a few slides ago. We were capturing trophic relationship, the white solid arrows, recycling relationship, the dashed one, and respiratory pathways, the dotted ones. And just to give you an idea of the additional complexity of this model, this is what it looks like. Lots of different processes being captured, from photosynthesis to soil nutrient leaching to herbivory, predation, and the recycling mediated by all of these animals. And in order to explore how different compositions of the ecosystem, with like either just soil and plants, or soil plants and herbivores, or soil plants, herbivores, and carnivores, shaped the ability of the ecosystem to capture carbon, we looked at net ecosystem carbon balance, which is a metric of how much carbon is either captured or outputted by the ecosystem. Now, generally, net ecosystem carbon balance is measured as as gross primary productivity minus ecosystem respiration minus lateral fluxes. So photosynthesis, essentially, that's gross primary productivity. Ecosystem respiration is all of the different respiration, whether it's predator, herbivore, plant, or soil. And then lateral fluxes, which are essentially the inputs and outputs, the inorganic ones. However, this formulation, which is the more used one, doesn't really account for the indirect effects that animals can have on an ecosystem through their behavior that is not herbivory or predation, so recycling, but also movement, trampling, giving birth and leaving birthing material on the ground that's very rich in nutrients and will get decomposed, will change what's happening in the soil there. So what we did was we took this equation for net ecosystem carbon balance, and we changed it a little bit. We updated it, and we estimated net ecosystem carbon balance for the purposes of this model as the algebraic sum of net primary productivity, so photosynthesis minus respiration, plus net heterotrophic productivity, which is essentially a way of saying we're taking the net production of the system and then we sum it to the algebraic sum of the net productivity of the predator compartment, the net productivity of the herbivore compartment, and the net productivity of the soil compartment. And that will give us an idea of not only how much carbon is captured or released by the system, but also of the effects, if any, that animals have on this ability of the ecosystem to capture carbon. So again, that can become a very long and convoluted mathematical equation. You probably are all not afraid of those, but I am. So I had Mathematica do it for me. And then once we had the new equation for net ecosystem carbon balance, we took our model and we ran several simulations comparing this base model to a model that included only herbivores, no predators, and a model that included only soil and plants, which was our benchmark. Now, in terms of what result did we get, notice that I had to move around the order of the models at the top. So now the soil and plant model without no animals, our benchmark is on the left. The herbivore-only model is still in the middle, and the predator model is on the right. What we found was that when we include animals, whether it's just herbivores or herbivores and predators, our toy ecosystem was able to capture, on average, 2 orders of magnitude, if not 3 orders of magnitude more carbon than the ecosystem that didn't include any animals. And that's the difference between the median, the thick line in each of these boxes. Now, the difference that you see between the model that includes only herbivores and the model that includes herbivores and predators is, sorry, is probably due to the trophic cascade effects of the predators, the suppression of herbivory and the release of plants. But this is still, on average, 2 orders of magnitude more of what we see when we don't have any animals. Now, I've been talking a lot about models and theoretical stuff. We're testing these results that we're getting from the model. We're trying to see if the real world agrees with how our model is saying, or whether we need to go and change our model. So we're testing this model of carbon capture mediated by animals in several different ecosystems. We have an experiment in the South Carpathian with European bison, that's on the left, with puma and guanacos in the Patagonian, in the Chilean Patagonia, in the middle. In the Yucatan of Mexico, we're testing a four species system with jaguar, pumas, white-tailed deers, and peccaries. And in Croatia, we're also testing a system with wild horses and toros, which are species of a little bit more wild cows, essentially. I just want to talk very briefly about some of the preliminary results we got for our case study with the bison. Our model performed acceptably well. We had results that showed, consistent with what our modeling was telling us, an increase in 2 orders of magnitude in the ability of the ecosystem to capture carbon when the bison are present compared to when they're absent. And to put that into perspective, it's about the same amount of carbon that is released in the atmosphere by about 2 million cars every year. And again, as a way to put it into perspective, the whole country of Romania has about 8 million cars. So by reintroducing, protecting, restoring systems to have viable bison population in that part of the world, we could potentially allow ecosystem to capture more carbon, enough to take out from the atmosphere carbon dioxide equal to what 2 million cars put out every year. So just briefly conclusion, then I think I'm done. I hope I convinced you today that animals can be key players in biochemical cycles. This is still an active area of research. It's a fairly new field of research as well, so we're still figuring it out. Animal movement can shape ecosystem functioning across landscape at local and regional scales, and animals can exert control on an ecosystem ability to capture carbon, which leads me to believe that animals could be allies in tackling climate change, and that studying how they shape the world could help us in designing new, possibly more effective nature-based climate change solutions than just planting trees and waiting for 6, 7, 8 decades before they start to do actually something. And I want to just help and acknowledge my collaborators, my supervisor, Oz Schmitz at Yale University, my PhD advisor, Sean LaRue at Memorial University, and my friends and colleagues at Yale and at Memorial, and the funders. Thank you.
Thank you, Matteo. And I think we're unfortunately out of time for questions, but I do encourage people in the audience to submit questions through the Q&A section, through the chat. And Matteo, please feel free to look over those questions and answer them as they come in.
And sorry for
so I'll pass over the space to Jerome.
So what I want to do is spend about 10 minutes talking about the math and the biology behind the model that I'm studying, and then spend the rest of my time, about the other 10 minutes, in an actual mathematical workbook or a notebook that y'all have, and we can just execute it together as we go. Let's see if I can get this to work. Wonderful. So let me point out, this work is a collaborative effort. Our research team is funded by the National Science Foundation. We definitely appreciate their support. It's composed of a math modeling group headed by Shivaji at UNCG, myself at AUM. We also work with Jim Cronin's lab at LSU. So let me start with, what is a density area relationship? This is basically just the relationship between the area of a patch and population density. Why do we care? Well, in terms of conservation, we want to know what's better. This is an ongoing debate in ecology. Should we take all the area that we have and put it in one large patch or several small? If we wanted to use density area relationship DAR in order to answer that, then a negative relationship, in other words, larger area meaning less density, would lead us to believe we need several small patches. However, if we have a positive relationship, then larger area means higher density. We want to go with a single large patch. Well, DAR's been well described for a lot of different animal taxa. In fact, empirical studies have found positive, negative, neutral, and even weird hump-shaped DAR within the same species, sometimes even within the same study. And so this is very interesting. We still don't, as ecologists, don't really have a good understanding of the mechanistic causes behind such variability. And so the theory that's often cited would be island biogeography or resource concentration or density compensation. But these theories really don't give much credence to why we would expect to see something like a hump-shaped DAR, which has been seen empirically. All right, so let me hone in on one particular mechanism here called predator-induced immigration. Now, so it's been a lot of work done. I'm just going to give one or two references here about looking at indirect effects versus direct effects of predation. So indirect effects oftentimes include this group called trait-mediated effects. And these things have been shown empirically as well as with models that they can really affect prey population. So the predator, not just the direct eating of the prey, but the presence of the predator can change prey habit, movement patterns, and foraging and all kinds of things. In particular, some studies have even shown that these indirect effects are oftentimes greater, have a greater effect than the direct density-mediated effects. One such example is by Cronin and others, where they looked at a system where they had several spiders acting as predators. They had some plant hoppers acting as prey. And what they found was as the spider density increased, it actually changed the plant hopper's movement behavior. And when they got to an edge of a patch, they were more likely to leave the patch in the presence of predators. They called this predator-induced immigration. Now, there's been a lot of empirical work that's tried to isolate these direct effects from the indirect effects. So Schmitz and others in 1998 looked at a very particular example where they took spiders and they glued their mouth parts together so that the spider could be present with the prey, grasshoppers, but spiders, the predator, couldn't eat the prey. And so they were able to, in an empirical system, separate the two effects. And so we really found that very interesting. Now, what I want to talk about today is how will predator-induced immigration, PIE, affect prey population dynamics, number one, but most importantly, how would it affect the density area relationship of the prey? So to date, very little had been done in terms of how would predator-induced immigration affect such things. All right, before I get to a model, let me give a context for this. We're also thinking about this at the landscape level. So we still, as ecologists, mathematicians, we don't really fully understand the relationship between persistence of a population that's living in a patch in a landscape with its patch size versus the patch geometry versus the patch matrix. As a mathematician, matrix is an unfortunate word, but in ecology, I believe this is most often described as this area around a good patch of habitat. So we're not fully able to describe the relationship between persistence and these things. So what I'm going to do today is I'm going to take the geometry part out of this. So I'm going to define a patch that's either in one or two dimensions. It's going to have a unit length or area, depending on the dimension. I'm then going to scale that patch coordinate-wise by a patch size parameter L. In this way, I can increase the patch size maintaining the shape or decrease the patch size also maintaining the shape. So this is going to make my job a little bit easier as a modeler. I can take the geometry, although it'll still be in the results, it's not a parameter that I have to change. So let me look at the model. So I've got a single patch surrounded by a hostile matrix. At every location in the patch, we're going to model predator density. Let's just think spiders for the time being, and prey density. Let's think grasshoppers. Here's the model. I don't have time in 20 minutes to go through all the details of this model. Let me just give you some highlights here. First and foremost, we are assuming mathematically that we have essentially glued the mouth parts of the predators. They are not allowed to eat the prey. So you'll notice for the reaction term in both of these reaction diffusion equations, there is just logistic growth for both. So we've kind of isolated in the same way that the Schmitz paper did. We've isolated the effects of direct density effects versus indirect. There is a random movement component here that's part of the diffusion term of these two equations. We're assuming an unbiased random walk in the patch as well as in the matrix. We are allowing this interface at the patch and matrix boundary here to, well, there's a lot of things going on here, but for sake of time, let me just say we've got two boundary conditions here in the model that will model this interface where animals are able to move into the matrix, experience hostility out here, maybe come back in through a random walk. The predator, U, is completely independent of the prey. So we're only focusing on the connection between the two in the prey's boundary condition. In other words, as the number of predators increases, the flow out of the prey increases. This is this predator-induced immigration aspect. Let me point out a few parameters here. This model has been non-dimensionalized, so the parameters here are not exactly what they would correspond to in ecology, but you can pretty much think of lambda as being patch size squared, or area. You can think of gamma 1 and gamma 2 being measures of how hostile the matrix is. In other words, like a death rate of the predator versus the prey. The beta here is what I call the trait-mediated behavior strength. This is a parameter that describes the sensitivity of the prey to the presence of predators. There's another parameter R here, but we'll gloss over that for the time being. Wonderful. So how will we compute the DAR? Well, we're going to fix the model parameters. We're going to choose an average patch geometry. We'll then assume the population density is near its equilibrium. We'll vary the patch size parameter L, and then compute the average density in the patch predicted by our model. So for our predator-prey system, what we would do is we would take a patch size. We would take 1 over either the length or the area of the patch. We would integrate the density. This would give me the average density in the patch. But remember, the way I've scaled the model, this patch omega has a unit length or area, so we can drop that all together. So I've got a nice formula here to compute the density area relationship. A similar formula would do the same for the prey. This is where Mathematica will come into play. We can actually get our hands on what these curves look like. And so for this project, we use Mathematica 13, but I know 14 has been recently released. Let me give some analytical results first, and then I will close out the PowerPoint and go to the notebook and actually play with Mathematica to create some of these density area relationships. When that sensitivity of prey to predators is small, then what we would expect to see is the following relationship. On the x-axis, I have predator density, think spiders. On the y-axis, I have prey immigration rate. When the sensitivity parameter is low, then there's a very small increase as the predator density goes up. We go from about 50% immigration rate, which would be random, up to maybe around 60, 65%, but it's a very slow increase. With that, we're able to prove mathematically the following scenarios. So what do we have here? It's a lot shown here. On the y-axis, I have patch size squared. So for larger lambda values, bigger patches. Smaller lambda, smaller patches. I've got at the top the different scenarios: unconditional extinction. You've got U can invade a patch, but V cannot. Coexistence. In blue, we have the predator here. That's their range of patch sizes where they're able to colonize or invade an empty patch. In kind of the pinkish color, the prey, or you could think grasshoppers, they have a smaller minimum patch size, so they're able to colonize a much larger range of patches. Let me give you an idea of what the results would look like. If you took a large patch in the coexistence range, then we would expect, if the prey came into the patch, the predators are also able to colonize the same patch, and at this level, there would be coexistence. Our model would predict coexistence for such a patch size. If I reduce the patch size some, we still have coexistence. I can further reduce the patch size. I'm still in the coexistence regime. Even though the patch is smaller, coexistence is still predicted. If we drop below this minimum patch size for the predator, then the prey can colonize the patch, but the predator cannot. If we take a patch that's too small, neither species is able to colonize or dwell in this particular patch. Let's look at another case. What if we increase the sensitivity of the prey to the predators? In the dotted curve, we've got the old case when beta was small. Now we've got in the solid black curve a much steeper response. As the predator density goes up, the prey immigration rate goes up even higher. Result-wise, everything is mostly the same. Large patches still give us coexistence. Even though the patch size is going down as I go across, we still have coexistence. We still have this range where only the prey is able to colonize the patch, the predator is not. if the patch is too small, nobody can colonize. What is very interesting here is analytically, we can see evidence of a nonlinear density area relationship. In fact, it should be hump-shaped. We don't know exactly what it looks like, but analytically, we can see there should be this hump-shaped DAR. Let me look at one more case, and then we'll go to the mathematical book. If that beta is very high, so here's the low case in dashed, the medium case in dotted, the high case in solid. So now any small increase in the predator density causing a dramatic increase in the prey immigration rate. What is predicted to happen by our model? First and foremost, for large patches, we still see coexistence. But interestingly, for intermediate range patches, we get an exclusion of the prey. In other words, prey are able to colonize the patch. But as soon as the predator invades, we end up with the predator excluding the prey. It's a trait-mediated exclusion. Remember, there is no direct effect in this model. This exclusion is solely from the indirect effects. So we were really excited to see that. But then interestingly, if you get even smaller patches, all of a sudden the prey can come back and you get coexistence. Once again, we have for this small range of patches that the prey can inhabit the patch, but not the predator. Finally, if the patch is too small, everybody dies. The most important thing I want to point out to you is, once again, a hump-shaped DAR is predicted. And so this is where we really want to go to Mathematica and see if we can look at what this curve is. So let me hit escape. Let me make sure you all can see my Mathematica document. So if you want to pull it up in the audience and go along with me, you are welcome to. So all of this is published in a paper in the Journal of Math Biology. I've got a link there for you. There are some notes here I'm not going to go over for sake of time. I will point out this notebook is for a one-dimensional patch, an interval with patch size L. So the first thing you're going to do is run the setup code. This will suppress some error messages. It will give us an estimate of minimum patch size. And then we will move on, and I'll talk very briefly about how we're going to do this numerically. So we have this boundary value problem. Let's just take the predator by itself. We have a boundary value problem for a steady state that we're trying to solve numerically. We're going to use a shooting method. Parametric ND solve can automatically do a shooting method. It's a beautiful method. However, for our particular problem, there were some issues with stiffness. And so we had to kind of go about that a different way. So what we're going to do is convert this boundary value problem to an initial value problem. We've got the boundary condition at 0, but at instead of putting on the other boundary condition at 1, we're going to specify an initial value for u at 0. We'll call that q. The point then is we're going to use parametric ND solve with the initial value problem and try to get a q value that will make this numerical solution also solve the other boundary conditions. Very standard method in solving these problems. Again, because of stiffness, we had a lot of trouble figuring out what initial q values to pick. If you pick the wrong q's, the method kind of struggles because the problem becomes too stiff. So we pulled in another method called a time map analysis. I don't want to go into too many details here, but I'll show you in a minute how we can use this time map analysis to give us just the right q values given a patch size to solve this boundary value problem. Let me give you a picture, just in case you don't know much about numerical mathematics. That's fine. Here is the spatial location in the patch, x. Here's the density scaled according to carrying capacity. We pick a starting value at 0. We have forced the boundary condition in terms of the slope to match exactly what we want. With this choice of q, we then follow the numerical solution and then see where we land at 1. If at 1, the boundary condition up here is also satisfied, we have found a solution to our boundary value problem. That's basically the idea. Wonderful. So step 1. We are going to use this time map analysis. For sake of time, I'm not going to go into the details. I've put a link here to a paper where we proved this and maybe talk more about that. Let me talk about some notation. For the predator, Pu will be its maximum density. Qu will be its minimum. For the prey, Pv will be its maximum density. Qv will be the minimum. For our G, we can prove that these solutions will always be symmetric about 1/2. So we don't have to really worry too much about that. So I need the reaction terms, they're primitive, the G that was in my original results. From the time map analysis, I need to solve this cubic equation. So using Mathematica's beautiful salvation tools, we can get in here and we can find this is a cubic, we can solve it exactly and come up with the 3 roots that we could have for a Q. So given a maximum value, these formulas will give me the minimum for the predator. I need to do the same thing for prey. So there's a different equation that I'm solving for that. I can use Mathematica once again to give me those exact roots. So we've done that. Let's run that cell. If we're successful, it should say setup loaded. The final couple of equations are also from the time map analysis. They give me a way to match the maximum value, the minimum value with the appropriate patch size. So I've got all the formulas that we need. So in the next cell, we are going to specify the matrix hostilities. I'll put these in here according to what they were in the paper. The beta values, which are already talked about, we'll start with 1. P-num is the number of bifurcation points we want. Accuracy here is listed. We'll leave all that as I put it, and we will run the cell. It's going to take around 30 seconds or so to run on my laptop. So while it's doing that, let me just briefly talk through the code. We are looping over the maximum value P, starting with the predator independent of the prey. We have 3 possible minimum values that are given here by the formulas above. We need to test to make sure that they are real. So the imaginary part needs to be small. We need to make sure that these values are bigger than 0. In ecology, negative densities don't make sense. In math, we don't care. But in ecology, they get worried. You start talking about negative animals. We need to make sure that the maximum, this is supposed to be the minimum, by the way, is below the maximum value P. So we're doing these checks to see which of these Q values is the correct one. We go through this loop and we store our answers in V0 values in the following way: max, accuracy, min, and the corresponding patch size, the V lambda. So that took about 40 seconds to do. We also have to do the same thing that was for the predator. We have to do the same thing for the prey, 0V. For the prey, in the presence of the predator, it's a little bit more complicated for now. Let me not talk about it and just show you some results. So what we are going to do, the previous code gave me those shooting values, the Qs. So all we've got to do now is use parametric ND solve, put in our appropriate shooting value, and we are guaranteed by the time map analysis to have our solutions. So we're going to run this cell that will load the parametric ND solve code. We are now going to loop through the lambda values, which is like patch size. We're going to extract the lambda, the shooting value. We're going to numerically integrate the density in the patch. That will give us the average density. We're going to pair that with patch size, average density, and create a whole list of these to plot. So we do that for the predator without the prey without the predator, and then the prey in the presence of the predator. We do all 3. We're going to run this code. It will go very quickly and immediately give us some results. So here we go. If we had a drum roll, we would play it right now. So what I have here on the x-axis, patch size squared, think area. On the y-axis, average density. Each of these curves is a DAR, a density area relationship. Blue represents the predator without the prey. Remember, the predator is independent, completely independent. So it really doesn't matter if the prey is there or not. The red curve represents the prey, but in the absence of the predator. And so you can see as the patch size goes smaller, the prey in the absence of the predator goes down, as you would expect. That's its average density in the patch. The purple here represents the prey's density in the presence of the predator. And so all of those do exactly what we would expect. This is for the case when the sensitivity of the prey to the predator is somewhat low. All right, great. Let me do this final code here. We'll go back and do another case. So in this final step, we're actually going to visualize the DAR and the actual density profiles and try to understand what's going on, what's behind these particular profiles. And so let's run this code. If it's correct, you'll end up with a beautiful manipulate. One of the things I just absolutely love about Mathematica. At the top, you have a parameter that represents the patch size squared. You could think area. On the left, we have our density versus on the y-axis, patch size or area on the x-axis. That's our DAR curve. On the right panel, we have the actual density distributions. We've got the patch location in X. We've got the actual density here. So these are the solutions that are given to us by parametric ND solve. Again, red is predator. Blue is predator. let's see, red here is prey in the absence of predator. Purple is prey with the predator's effects. If you take a very large patch size, you see that these densities are almost at 100% of their respective carrying capacities. As we decrease patch size, you can see these density profiles begin to decrease. We're running out of space. The animals are encountering the hostile matrix more and more and more. That's costing them in terms of population. The predator in blue will actually come to its minimum patch size quicker, and you will see it go down. Its density level will go down to 0. In this range where the predator is not able to colonize a patch, it's not present. Only the prey is able to colonize a patch. If the patch size goes too small, everybody dies. So let me go back. Let's look at a different case here. If you'll go back to the step, let's see, step 1, where we specified our parameters. We're going to change the beta to 20. We want to see this hump-shaped density area relationship. So run that code. On my laptop, it takes around 30 seconds or so. So we're going to let that run. I'm going to scroll down here to the bottom while this is running. Let's see, the last one took about 39 seconds. Again, I'm running this on a laptop. If I had a bigger workstation, it would probably go faster. Let's see. Oh, 25 seconds. Very nice. So now I'm going to run the next cell. You can run these parametric ND solves again. You will then run the cell where we're going to create the DAR curves. You will see in this beautiful picture, you can see in the purple, the hump-shaped DAR curve. So as we start from large patch sizes and we decrease the lambda, which is patch size squared, you can see for purple, that's the prey. In the presence of the predator, their DAR curve is decreasing, but then all of a sudden shoots back up, peaks, and then comes back down in the red. So we get a hump-shaped DAR curve, and you'll notice here the sensitivity to predators is much higher. So let's go to the final cell and look at the manipulate and see if we can understand what's really going on here. So I'm going to start with very large patch sizes. For a very large patch size, as I begin to decrease the patch size, we can see the density coming down. Remember, purple is prey with the predator. Blue is just the predator, independent of everything else. What you will notice is, as the patch size is going down, the densities are going down. That's what we would expect. But something interesting happens here as we start getting close to around 5 for lambda. You'll notice that the blue curve begins to decrease much faster than the purple curve. It's a blue curve. The predator density is beginning to decrease much, much faster. And in fact, because it's decreasing faster, that causes the prey density curve to actually come back up. As the area is going down, the prey curve is now going up. So what it is it's a release from predation. The indirect effects of the predator on the prey are being released because the predator density is going down to 0. That's what's causing this hump-shaped DAR. And it's these beautiful graphs in Mathematica that let us dive in here and see exactly what's going on. And so this is very interesting. What it implies is that there's kind of an intermediate value here for patch size where the prey's density would be maximized in the presence of the predator. Okay, for sake of time, let me jump back over to the presentation. Let me just throw up some conclusions and leave it there for questions. Let's see if I can get this to come back up. There we go. So just very quickly, we have limited theory to explain why the DAR would be higher for intermediate areas. It's really unclear to this point how trait-mediated effects such as PIE would affect the DAR. So what we did was look at models that are equivalent to gluing the mouth parts of the predator, very much what Schmitz did in 1998, to decouple the direct and indirect effects. We employed this with very landscape level attributes. What we found is we can get hump-shaped DAR when the trait-mediated response is sufficiently strong. Wolfram Mathematica beautifully allowed us to create these curves so we can see exactly what's going on from a mechanistic point of view. For ecologists, I think what this suggests is we should really consider these indirect effects of predation. and there may be no one solution for species conservation using DAMR. So let me stop there.
Thank you, Jerome. That was fascinating. I want to tie this back to the very beginning of your presentation, where you were talking about the choice of partitioning of protected areas, for example, in conservation. And I was wondering, just if in a minute or so, you could talk a little bit about what this research, how this research might inform those decisions in the process, in the context of conservation strategies, for example.
Yeah, great question. So there's two big aspects to our research. We're looking at things at a landscape level. So you've got to worry about the patch size as well as its geometry, and then how hostile this kind of bad area called matrix is surrounding the patch. So in some cases, it may be better to spend money and effort restoring the matrix. You may get more bang for your buck, kind of improving the condition around the patch rather than trying to restore the patch. Another aspect of this is these mathematical computations, especially using Mathematica, can really dive into the different geometries of a patch and can show you for a given patch in the real world, you can take its geometry, feed it into the model, look at minimum patch size. You could understand better what role geometry plays. Maybe we need to work on the shape of the patch more so than the size of the patch. I don't know if that helps answer your question any.
Thank you very much. I think this is helpful. I think so. In the amount of time that we have left, I'd like to just have a short sort of closing section and discussion. And I'd like to give an opportunity to all of the speakers, maybe in order of presentation, to answer a couple additional questions, maybe from the audience, but also I have a couple questions to get us started. So I'd like to start with. Well, we've heard some really fascinating topics presented today, and I see a lot of overlap between some of the materials from our presenters. I'd like to ask you, based on the materials that have been presented today, are there potential ways that you might see the material aspects of the materials that have been presented by other presenters incorporated into future work, into future research that you might into your future research? Perhaps Gareth, you can start. Yes.
Yes. So it's interesting, especially given what Jerome was talking about. I mean, when I began my current position, I was interested in metapopulation dynamics and the dynamics of populations sort of in patchy environments in general. That was my background for my PhD, looking at island biogeography, patterns and things like that. And then one thing I began to realize is that the part about dispersal, right, in all the models, there's a lot of existing models for sort of, population dynamics on patches, but the part about dispersal was the least understood part. And most of these models had at their core a ridiculously simple assumption that assumed that, for instance, individuals blow around like leaves in the wind, like some sort of more or less random dispersal model, which anyone who's watched almost all organisms, they don't behave like that, right? They generally power themselves along and they know where they're going. There are some exceptions, but most large organisms are pretty much in control of their movements. And so I ended up switching from thinking about the patch dynamics itself to thinking about movements, because I kind of wanted to get more directly at that aspect of how animals choose to move about, how they cross boundaries, right? And therefore how things like the orientation and the shape of patches really affects the dynamics of populations on them. So, what Jerome is talking about the importance of like patch shape. And I remember playing around with different models of animal movements sort of between patches and thinking about, patches that are sort of long and thin versus round, and how much animals perceive of where other patches are, something you might call the viewscape of animals and their sort of perceptual abilities. That's very much what, and then eventually I ended up thinking about elephants moving around landscapes, because you you start down a path and then you sort of just keep going and end up working on animal movements. But it all started with thinking about the dynamics of populations on patchy environments and the importance of the geography and the geometry of those patches.
I found really interesting the way that you were modeling the movement of animals in your landscape in Etosha, Gareth, and the incorporation of competition in trophic levels in Gige stock, and the indirect predation effects. And I can see all of those integrating all of those different aspects in ecosystem models in the future. Actually, I am currently thinking about how to diversify the trophic compartments that make up the structure of the ecosystem models that I use by integrating different characteristics of different species. So things like functional trait, the ways that animal forage, or the ways the animal move, or other features of the animals that like set different species apart and maybe shape the functions that they perform in ecosystems. So yeah, definitely it would be interesting to explore those things, and I have lots of new ideas to play around in my mind after listening to all of the talk today.
I also learned from Matthew's talk that there's a more intrinsic relationship between herbivores and plants. They're not just like killing each other like what I modeled. So also I would love to like study how like carbon source and nitrate source and all type of like nutrients, including these type of models. So that's a good future direction I'll probably work on.
Yeah, I mean, my thought would be, and this is coming from, I'm a mathematician, I'm not an ecologist. I enjoyed really the different aspects of the talks, and what struck me was the different questions that were being answered by looking at different scales. I mean, that's really what it came down to in terms of the models. Some of them, like Garrett's talk, was at a very fine scale, I would say, movement-wise, whereas the models I'm using make some assumptions like random dispersal, because we're trying to scale up from those individual movement decisions, trying to get at a higher level what's happening at something like the area versus density curve. And so I was just really struck by the scale at which each of these talks were studying models to answer biological and ecological questions. I just thoroughly enjoyed it.
I was just going to say that for those of us who study or analyze sort of empirical data from the field, the changing technologies mean that we have moved scale just because we can. So when I first started looking at elephant movements, the collars were taking a fix every 6 hours, right? And now it's every 15 minutes, and actually newer collars take a fix, like every minute. We have some data from the collars that have been tried out on goats where the fix is every second, right? And that's just crazy. And then the satellite imagery and the resolution at which you can analyze the landscape has gone up by like orders of magnitude as well. It's almost overwhelming to sort of keep up with those things, but it means that working with empirical data, you can sort of move from we used to ask like very broad questions, right, as well, because that was the only way that they could be tested. But now the data are there to sort of take it down, to sort of finer and finer scales. So I think that's a lot of what's kind of driving some of the movement between scales as well.
Well, I just want to thank you all again for accepting to present for this event. Your contributions are what make the events like these possible. And thank you as well to our great audience members and their great questions. I really appreciate. I feel that this has been a very successful event, and I just want to convey my thanks to all of you.